going to introduce Professor Eija Pirinen, who's speaking from the University of Helsinki. Eija used to be my boss at one time, because as well as having a contract in Sweden, I also had one with the University of Helsinki. Anyway, I discovered that she was doing such great mitochondrial research that I asked her to come and speak today. So I think this is going to be a terrific presentation. So Eija, over to you.
Thank you, Rachel. Can you hear me properly? Can you raise the thumbs up? Or...
Yes, we can hear you.
You can hear. Okay, thank you, guys. So greetings from Helsinki. It's my great pleasure to today tell you about our mitochondria-related studies on mitochondrial myopathy and obesity. My research group is located in Helsinki, and here you can see our university campus near the city center. My research group is working on the development of new treatment options for mitochondrial dysfunction. And here you can see the currently available approaches to cure dysfunctional mitochondria. The easiest option is, of course, the improvements in lifestyle habits, healthy diet and exercise. But additionally, for example, gene therapy and mitochondrial replacement therapy are used for mitochondrial disorders in which mitochondrial dysfunction is caused by genetic mutations. And the third commonly used option to cure mitochondria is treatment with pharmacological compounds. But the current evidence suggests that nutritional interventions can delay and even prevent the development of diseases with mitochondrial defects. And therefore, I think that future studies should focus on the generation of cost-effective nutritional therapies for the treatment and prevention of diseases with mitochondrial dysfunction. And my group has been investigating vitamin B3 forms, which are found in food ingredients. So they are naturally occurring compounds. And there are actually three forms of vitamin B3, niacin, also known as nicotinic acid, nicotinamide, and nicotinamide riboside. Vitamin B3 forms are water-soluble. The excess can be excreted in urine through the kidney. And here you can see good sources of vitamin B3, fish, meat, milk products, whole grain, eggs, and green leafy vegetables, nuts, and yeast. The recommended daily allowance for vitamin B3 is 15 to 20 milligrams per day for adults to prevent vitamin B3 deficiency. And the deficiency can cause a disease called pellagra, which is characterized by dermatitis, diarrhea, and dementia. Niacin was discovered by Casimir Funk, who isolated niacin from rice polishings in 1910. And niacin has been used in high doses for the treatment of dyslipidemia over 50 years. But as a side effect, niacin can cause cutaneous flushing as it activates niacin receptor GPR109A. Niacin is not available on the market in Europe in high doses like in US. Nicotinamide was discovered by Konrad Elvehjem, who purified it from liver in 1937. Nicotinamide doesn't improve circulating lipid values like niacin, but it doesn't activate GPR109A, the niacin receptor, and it doesn't therefore cause the cutaneous flushing. But nicotinamide has been used for the treatment of pellagra and cutaneous inflammation. Nicotinamide is not available as high doses on the market in Europe. Nicotinamide riboside, NR, was found in milkweed by Charles Brenner in 2004. It doesn't cause cutaneous flushing as it doesn't activate the niacin receptor like nicotinamide. And interestingly, in animal studies, nicotinamide riboside has lowered cholesterol levels and improved the metabolic health and protected from the diet-induced obesity. NR is sold as a compound called niacin in US. First clinical studies are ongoing in humans, and NR has been observed to be safe and well tolerated. But how these vitamin B3 forms are utilized in our cells, how they are metabolized. So it's a well-known fact now that all these forms are needed for the biosynthesis of NAD, which is the important cofactor for mitochondria energy production. So all these forms, NR, nicotinamide, and niacin, are converted to NAD via distinct biosynthetic routes in our cells. But what is the role of NAD in our cellular energy metabolism? Otto Warburg discovered in 1936 that NAD is involved in redox reactions in which it acts as an oxidizing agent. It accepts a hydrogen ion to form NADH. And like Rachel already explained to you, NAD-NADH redox couple is important for many mitochondrial reactions in our cells. This redox couple is required for enzymatic reactions in TCA cycle, but also for oxidative phosphorylation, which creates ATP for our cells. So many mitochondria reactions are dependent on NAD-NADH redox couple. But how this NAD biosynthesis is regulated? In humans, this is unfortunately still relatively unknown, but based on the animal studies, aging and high-fat feeding and sedentary lifestyle decrease vitamin B3 dependent NAD biosynthesis, while fasting, calorie limitation, and exercise increase NAD biosynthesis. Now, based on the animal studies, it's also known that in many chronic diseases, there are disturbed NAD biosynthesis and NAD deficiency in different organs. And as an example, NAD deficiency has been observed in mitochondrial myopathies, in muscle dystrophies, but also in metabolic diseases like obesity, fatty liver disease, and type 2 diabetes, but also in neurodegenerative diseases. But what are the consequences of NAD depletion? As NAD is required for the mitochondria energy production, it's logical that NAD deficiency leads to mitochondria dysfunction. But also NAD has an important role in many other cellular processes, and consequences can be severe for many cellular processes. Like, for example, NAD deficiency can lead to DNA damage, loss of proteostasis, altered cellular communication, and increased cellular senescence. But how to boost intracellular NAD levels? Of course, again, healthy diet and exercise are good options. But now, based on the animal studies, it's known that supplementation with vitamin B3 forms can efficiently boost intracellular NAD levels in different organs. And in the next part of my presentation, I will show you our data from clinical trial in which we supplemented mitochondrial myopathy patients with vitamin B3 niacin. But first, I would like to introduce you mitochondrial myopathies. So they belong to groups of diseases called mitochondrial disorders. And these are diseases, rare diseases with amazing clinical variability. And these diseases are caused mitochondrial dysfunction. And in many cases, the mitochondrial dysfunction is caused by genetic mutation in nuclear genes or deletions in mitochondrial DNA. And mitochondrial dysfunction in these diseases can affect several all. And like I explained to you already, I don't know whether you heard this, but I explained that this disease is characterized by progressive weakness and atrophy of external eye muscles. So this leads to droopy eyelids. And typically the patients demonstrate muscle weakness and exercise intolerance. And commonly this disease is caused by sporadic single mitochondrial DNA deletions or mutations in twinkle or pol gamma genes, which are involved in the maintenance of mitochondrial DNA. And the mitochondrial myopathy patients, they show mitochondrial DNA deletions in their muscle cells, muscle fibers, so they can be seen with these kind of smears, small faint bands when running agarose gel electrophoresis after PCR amplification. And also additionally, these patients typically demonstrate complex 4 negative succinate dehydrogenase positive abnormal muscle fibers. The abnormal muscle fibers are seen here with the blue color and light brown color. When I was postdoc in Johan Overk's lab, I was involved in 2 studies where we investigated whether nicotinamide riboside NR supplementation could alleviate the disease symptoms in 2 mouse models for mitochondrial myopathy. And indeed, we noticed that in both models, NR markedly delayed the disease progression. And when I returned back to Finland to continue my studies, together with Anu Suomalainen-Vartiovaara, we decided that based on these encouraging findings in mice, it's of importance to run a clinical trial with vitamin B3 in myopathy patients. And next I'm going to show you the key findings from this article published last year in Cell Metabolism. So aim of this clinical trial was to investigate whether vitamin B3 supplementation would delay the progression in mitochondrial myopathy patients like in mice. But we were also interested in studying whether mitochondrial myopathy patients show abnormalities in NAD metabolism. And here you can see the study design of our clinical trial. We decided to use niacin instead of nicotinamide riboside because at the time when we started the clinical trial, NR wasn't pure enough for the pure enough for human studies. And we used this kind of time-released, slow-release form of niacin to reduce the flushing incidence and severity of the flushing. And the dose was 1 gram per day. It was selected based on the earlier studies with niacin showing the beneficial effect on lipid profiles. And the treatment time differed a bit between patient and controls. In both cases, we supplemented study subjects with increasing dose of niacin. The treatment dose 1 gram per day was achieved after 3 months. And total treatment time for patients was 10 months and for controls 4 months. And we collected muscle biopsies, blood samples, And also we performed clinical evaluations of the study subjects at several time points. For patients, we performed these sample collections and clinical evaluations at baseline and after 4 and 10 months of niacin supplementation. But for controls, we performed these measurements and sample collections at baseline and after 4 months. This slide summarizes the characteristics of our study subjects. We managed to recruit 5 patients with mitochondrial myopathy. They all had droopy eyelids, proximal and central muscle weakness, and exercise intolerance. Age of the study subjects or patients varied from 17 to 70, and 4 out of 5 of the study subjects were females. We also recruited 2 age and gender matched healthy controls, meaning that we had 10 controls in total. First, we assessed whether mitochondrial myopathy patients showed abnormalities in their NAD levels in humans, both in diseased and in controls also. Next, we examined the effect of niacin on body composition, and we observed that body weight did not change upon niacin supplementation, but in controls, we observed a significant decrease in fat percentage, and also we noticed that muscle mass increased significantly both in controls and in patients. But strikingly, niacin supplementation decreased liver fat content and visceral fat content in patients after 10-month supplementation. In controls, there was also a trend for a slight decrease in subcutaneous fat amount. So these results showed that niacin can improve body composition, especially in mitochondrial myopathy patients who demonstrated, for example, hepatic steatosis at baseline. When we analyzed the energy expenditure of our study subjects, we noticed that there was a trend for increased basal metabolic rate upon niacin supplementation, both in controls and in patients. And therefore, the increase in oxidative metabolism and energy expenditure could at least partially explain why we observed beneficial effects on body composition. Niacin has been reported to increase circulating levels of adiponectin, which is adipokine secreted from white adipose tissue and known to stimulate fatty acid oxidation in the liver. And therefore, we analyzed the circulating levels of active form of adiponectin in our study subjects. And we noticed that there was a robust increase in the levels of adiponectin in both study groups, possibly also explaining the reduction seen in the liver fat content, especially in mitochondrial myopathy patients. As niacin has been shown to reduce efficiently lipid levels, we measured also circulating lipid species in our study subjects. As can be seen, niacin did not affect total cholesterol levels, but there was a significant increase in HDL2B particles, which are like anti-atherogenic HDL particles. In addition, niacin supplementation decreased harmful lipoprotein levels, apolipoprotein P levels, and there was also a significant decrease in LDL and triglyceride levels in patients. So niacin improved the lipid profiles in patients especially. Niacin has been reported to slightly increase insulin resistance, And additionally, niacin can be hepatotoxic with the doses over 3 grams per day. And therefore, we analyze glucose metabolism related parameters and liver function parameters. As can be seen, fasting glucose levels increased in all study participants, especially at the time point 4 months. And C-peptide and insulin levels were also significantly elevated in controls at this time point. So there was a slight increase in insulin resistance, but luckily, glucose-related hemoglobin levels, the marker for long-term glucose homeostasis, remained stable during the study, and these values remained within normal reference values. We did not notice any significant changes in liver function parameters, suggesting that the used dose 1 gram per day was not hepatotoxic. As niacin supplementation restored the NAD levels in patient muscles, we next asked whether this was translated into improvements in muscle histology and the disease markers. And interestingly, we noticed that after niacin supplementation, the number of sick abnormal COX-negative SDH-positive fibers was significantly reduced after 10 months of supplementation. And when we analyzed mitochondria-related parameters, we noticed that mitochondria oxidative capacity, analyzed as a complex 4 activity, was significantly elevated in controls, but importantly also in patients after 10 months supplementation. And in line, we observed that mitochondria amount increased in both study groups. So these findings are in line with the preclinical studies showing that vitamin B3 supplementation can increase mitochondria biogenesis in muscle. Next, we studied whether improvements in mitochondria function was reflected in a patient's performance. And indeed, we noticed that exercise capacity was improved in patients as they were able to walk longer distances during the 6-minute walk test. And strikingly, we noticed that the niacin supplementation increased the strength of core muscles, back and abdominal muscles in patients. Additionally, we noticed that niacin supplementation improved the strength of upper and lower limbs also in patients, but also interestingly in controls. And these findings demonstrated that niacin has a potential to also restore or delay the muscle weakness progression in patients. We also investigated the effect of niacin supplementation on muscle global metabolic profiles using a metabolomics approach. And we observed that niacin supplementation shifted the global metabolic profile of patients towards controls. So here you can see with the pink color the metabolic profile of patients at baseline, and blue color, blue cloud, is the metaboloid profile after niacin. And here you can see on the right-hand side the metabolic profiles of controls. So clearly there is a shift towards controls. And we observed the many changes, for example, in amino acids and metabolites related to purine metabolism. Based on the questionnaires, patients' quality of life remained stable during the clinical trial. Niacin was well tolerated, but typical side effects like flushing, gastrointestinal irritation, skin problems like skin drying, and flatulence were reported by study subjects. 2 controls discontinued due to gastrointestinal irritation after 8 weeks of supplementation. we noticed that during the study, hemoglobin slightly decreased in all patients, but not in controls. And therefore, there is likely a need for hemoglobin monitoring in mitochondrial myopathy patients upon niacin supplementation. Based on these results, we can conclude that niacin can mitigate hepatic steatosis, increase muscle mitochondria oxidative capacity, improve muscle weakness, and decrease the number of sick muscle fibers in mitochondrial myopathy patients. And therefore, niacin supplementation is a novel and promising treatment option for human adult onset mitochondrial myopathies. But the optimal dose needs to be still established for these patients, and there is a potential risk for anemia in these patients upon niacin supplementation. Okay, let's move to my last part of my presentations. So I would like to tell you about our mitochondria-related studies on obesity. As you all know, obesity is a huge problem worldwide, and therefore effective treatment options are needed. And in obesity, there is a mitochondrial dysfunction observed in wide adipose tissue. And this defect in mitochondrial function is likely acquired, for example, via excess calorie intake and sedentary lifestyle, but there could be also other factors. And this slide summarizes known abnormalities in mitochondria-related markers. It's now known that the large adipocytes normally show lazy mitochondria with diminished oxidative capacity. There is a typically reduction in mitochondrial number, and the morphology of mitochondria is abnormal, and there are disturbances or abnormalities in the mitochondria quality control systems and turnover. Together with my collaborator Kirsi Pietiläinen, we have studied whether adipose tissue mitochondria capacity plays a role in the weight loss success. And we published 2018 that adipose tissue mitochondria capacity predicts weight loss success. So we observed that the higher the mitochondria mass and oxidative phosphorylation capacity in adipose tissue, the more successful the weight loss is in response to conservative weight loss treatment. And here the conservative weight loss treatment was based on the calorie limitation. But we also investigated in this study what is the effect of conservative weight loss on adipose tissue mitochondria oxidative capacity. We hypothesized that weight loss will improve mitochondria oxidative capacity. But let's go to the results soon. So we had two groups of obese subjects undergoing weight loss intervention, this calorie limitation-based weight loss intervention. One group successfully lost weight throughout the study, but this another group of obese subjects, they initially lost body weight, but after 6 months regained weight and fat mass. But interestingly, when we studied the adipose tissue oxidative phosphorylation, we observed that in both study groups, oxidative phosphorylation capacity surprisingly declined. So this was what we did not expect at all. And we realized that maybe this is kind of mechanism explaining why any individual easily gain weight and fat after weight loss interventions and different type of diets. And therefore, we think at this moment that there is a need to find a diagnostic tool to measure adipose tissue mitochondria function before any kind of weight loss intervention, and find a novel treatment options to revive adipose tissue mitochondria to support the weight loss interventions. But I would like to point out that not all weight loss interventions have a similar kind of impact on wide adipose tissue mitochondria. So my collaborators published last year that bariatric surgery actually improves mitochondria oxidative capacity in wide adipose tissue. So if we focus here, we can clearly see that bariatric surgery improved the expression of mitochondria-related pathways in wide adipose tissue, whereas conventional weight loss decreased the expression of these many interesting pathways in mitochondria. So bariatric surgery seems to have opposing effect on adipose tissue mitochondria than the conventional weight loss. And like I said, now there is a clearly need to find treatments and tools to activate wide adipose tissue mitochondria in obese subjects. And vitamin B3 form nicotinamide riboside, NR, is heavily studied at this moment in the context of obesity. And I was involved in one study showing that NR can really protect against high-fat diet-induced obesity and diabetes in mice. And now the clinical trials with NR are ongoing. And we have also completed recently one clinical trial in BMI discordant twins, and we are going to report our interesting findings soon. But to date, short-term NR supplementation has not been published to influence body composition or mitochondria glucose or lipid metabolism in humans. But we can conclude based on the obesity-related studies that conventional weight loss intervention suppresses adipose tissue mitochondria oxidative capacity, and this is probably adaptation and favoring the weight regain after dieting. But in contrast, bariatric surgery increases adipose tissue mitochondria oxidative capacity, leading to enhanced mitochondria energy consumption and energy dissipation. And we think that adipose tissue mitochondria profiling could be used as a prediction tool for weight loss success. And this tool could ease the access So lastly, I would like to mention that in future, it would be important to evaluate the prevalence of NAD depletion in mitochondria disorders and human diseases. And what is important in future is to optimize the vitamin B3 dose and treatment time in humans. And also we are interested in assessment of vitamin B3 rich diet on NAD and mitochondria homeostasis in humans. And it's really important to determine the mechanism via which bariatric surgery improves the mitochondria capacity in adipose tissue. And I believe that it's important to develop personalized therapies for obese subjects with a reduced mitochondria capacity. So this is my last slide showing my key collaborators. Thank you for your attention, and I'm really sorry about these technical problems.
Well, I'm sorry it was disrupted, but thank you very much indeed. Do we have any questions? We just have a few questions for you.
Thank you very much for the lecture. I just wanted to ask, do you give 1 gram in one dose or in split doses?
In the case of niacin, we gave 1 dose only in the evening with a light meal. With the nicotinamide riboside, we have split it that we give ½ gram in the morning, ½ gram in the evening.
Thank you.
I've got a question from one of the webinar viewers. Why do you think NAD plus depletion takes place in mitochondrial disease and chronic disorders?
So there seems to be well, there are studies showing that NAD biosynthesis is disturbed in many chronic diseases. In mitochondrial disorders, we also noticed that there were some abnormalities in NAD biosynthesis. But in the case of mitochondrial disorders, the NAD depletion could be caused by impaired complex 1 function. So OXPHOS is not properly functioning, and this leads to accumulation of NADH and reduction in the NAD levels. So there must be different mechanisms in the case of mitochondrial disorders leading to NAD depletion. And additionally, there are also enzymes, for example, needed for the DNA repair. They are called poly-ADP ribose polymerases. So they are consuming heavily NAD for the reactions they catalyze. So they are involved in the reactions leading to DNA repair. So like in the case of mitochondrial myopathy, PARP activity could be elevated as there is a need to repair the mtDNA.
Okay, thank you. Any more questions from the floor? We're just bringing the microphone.
Thank you very much, Dr. The Professor. Thinking about the pathology of myasthenia gravis, this one that we have come into mitochondrial myopathy. What do you think about that if you compare these two conditions? Can it lead to misdiagnosis as now mitochondrial myopathy is developing and coming out with very distinctive features and maybe markers or parameters which could really make us go back to review myasthenia gravis?
So what was your question? I did not hear properly. So how to diagnose this?
Hold the microphone a little further away. Make it a short, concise question.
I was asking the difference that could come up between myasthenia gravis, that's one of the, I think, pathology in muscles as compared to myopathy, mitochondrial myopathy. What do you think about the two conditions?
Did you get that, Eija? Oh, it's frozen.
Okay. That is the last time she'll freeze this afternoon.